Hello, my dear students. Welcome you all to Sri Jain Rank Zone classes. I'm your mentor and your friend for the subject biology, and I will be taking the zoology part that is in subjects. My dear students, in the last lecture we have started chemical coordination and integration. We discussed the introduction of endocrine glands, endocrine system, endocrinology, hormones, their chemical structure, their chemical properties. We have also discussed about hypothalamic hypophyseal stalk system. We discussed about different controlling hormones or controlling factors which are released by hypothalamus. Now I am going to talk about the classification of hormones on the basis of mode of action. So on the basis of mode of action, hormones are classified as quick acting hormones and delayed acting hormones. Quick acting hormones, these hormones initiate immediate response for their target cells. Their receptor is always located on outer surface of plasma membrane of target cell because they are large sized. Okay, as the receptors are large sized, they are always located on the outer membranal surface of plasma membrane of the target cell. Hormone receptor complex activates the membrane enzyme adenyl cyclase, which hydrolyzes ATP into cyclic adenosine monophosphate. So quick acting hormones, these hormones initiate immediate response from their target cells. Their receptor is always located on the outer surface of the plasma membrane because of high molecular weight. The hormone receptor complex activates the membrane enzyme adenyl cyclase, which further hydrolyzes adenosine triphosphate into cyclic AMP. The cyclic AMP acts as a secondary messenger. Cyclic AMP activates the inactive enzyme system by a cascade effect. So their mode of action is always second messenger hypothesis. So always quick acting hormones, they followed which type of hypothesis? Second messenger hypothesis. This includes proteinaceous hormone, peptide hormones, and amine hormones. So basically, the hormones which are made up of amino acids, or they have amine group, or they have peptide linkage, or they are proteinaceous in nature, they are polypeptidic in nature, they always follow quick acting hormones, or they act on the target cells having the receptors on the membrane, plasma membrane. Now, delayed acting hormones. These hormones initiate response after some time. They are small sized, and that is why they are diffusible. They can move inside the cell through the plasma membrane of their target cell. These bind their proteinaceous receptor present in the cytosol. They always operate through de novo synthesis of mRNA, messenger RNA, by activation of certain genes. So they basically forms a new synthesis of, or a new formation of messenger RNA for the activation of certain genes. Their mechanism of action is always mRNA hypothesis. They include steroidal hormones of testes, ovary, adrenal cortex. So the steroidal hormones always follow the delayed acting hormones. They are delayed acting hormones as they always act on the nucleus. They helps in the formation of new messenger RNA, which will further, you know, make a new gene, a certain gene, which are required for the effect of hormones. So all the steroidal hormones, they function in the category of delayed acting hormones, and all the proteinaceous hormones, they function under the category of quick acting hormones. Difference between hormone and enzymes. On the basis of chemistry, enzymes, they are always proteinaceous. Hormones may be proteinaceous or amine or steroids. Enzymes, their molecular weight, they are macromolecules with high molecular weight. Hormones, they have low molecular weight. Diffusibility of enzymes, they are non-diffusible through cell membrane. Hormones, they are diffusible through cell membrane. Enzymes, they act intracellularly or carried by some duct on the other side. They are generally carried by blood to the target organ. Enzymes always act as a biocatalyst and increase the rate of metabolic physiologic process. Hormones, they may be excitatory or inhibitory in their physiologic action. Enzymes, they catalyze reversible reactions. Hormones, they control the reactions which are not reversible. So hormones basically controls the reactions which are not reversible, and enzymes, they only catalyze reversible reactions. The rate of the reaction increase with increase in the concentration up to a particular limit, that is optimal concentration. Hormones deficiency or excess of hormone causes metabolic disorders and diseases. Enzymes, they act quickly. Hormones, they some are quick acting while some other are slow acting with a large lag period. Enzymes, they are not used in metabolic functions. Hormones, they are used in the metabolic functions. Okay. Enzymes, their concentration is not affected in the metabolic functions, while hormones, their concentration is affected in the metabolic function. So these are the differences between hormone and enzymes. Now, mechanism of hormone action. The hormones, they work in two ways. Either they work by two ways. On the cell surface, the molecules of hormones that are amino acid derivatives, peptides, or proteinaceous in nature are large and insoluble in lipid. They are insoluble in lipid and they cannot enter the target cell. Therefore, they act at the cell surface. They bind to the specific receptor molecules located on the surface of cell membrane. The hormone receptor complex may act in one of the two ways. So on the cell surface, the molecules of hormones, especially which are amino acid derivatives or peptides or proteinaceous in nature, they are large and insoluble in lipid. They cannot enter the target cell. Therefore, they act at the cell surface. They bind to the receptor molecules which are present on the cell surface or the surface of the cell membrane. The hormone receptor complex may act in one of the two ways. First way out is formation of cyclic AMP. Mechanism of formation of cyclic AMP was discovered by E. W. Sutherland in 1950. Very important. The hormone receptor complex causes the release of an enzyme adenyl cyclase from the receptor site. This enzyme hydrolyzes the ATP, break down the ATP into cyclic AMP, cyclic adenosine monophosphate. The cyclic AMP activates the existing enzyme system of the cell. This accelerates the metabolic reactions in the cell. The hormone is called the first messenger and cyclic AMP is called the second messenger. Adrenaline causes secretion of glucose from the liver cell. Okay, so adrenaline is the first messenger and cyclic AMP is the second messenger, which is basically increasing the secretion of glucose from the liver cell. So this is the formation of cyclic AMP, which is considered to be the first optimal mechanism of hormone, okay, which is present on the in which are acting on the receptors on the cell surface. This is the mechanism of hormone action on the cell surface. You can see hormone is the first messenger. These are the transmembrane receptor proteins which receive the hormones. Okay, it further activate the G protein, okay, and form the adenyl cyclase. It further hydrolyzes the ATP into cyclic AMP. The adenyl cyclase, it further catalyzes the conversion of ATP into cyclic AMP, which acts as the second messenger. It is further degraded by the phosphodiesterase enzyme. Cyclic AMP is further degraded by phosphodiesterase enzyme. Cyclic AMP activates protein kinases. It can alter the activities of metabolic enzymes. It can alter the permeability of plasma membrane. It can cause muscle contraction and relaxation. It can stimulate protein synthesis and breakdown of lipids. It can also alter the rate of secretion. So these protein kinases means the activated protein kinases, they can alter the activities of metabolic enzymes. They can alter the plasma membrane permeability. They can cause muscle contraction and relaxation. They can stimulate protein synthesis as well as the breakdown of lipids. It can also alter the rate of secretion. Now you can see the water soluble hormones are membrane insoluble. They bind to the membrane receptors. The binding activates the G protein. Okay. The activated G protein activates adenyl cyclase. Adenyl cyclase further conversion or convert or catalyze the hydrolysis of ATP into cyclic AMP. The secondary messenger in this pathway, the cyclic AMP further activates the protein kinases. Protein kinases phosphorylate, addition of phosphate group proteins in the cytoplasm. This activates these proteins, allowing them to alter the cellular activity. So this is the mechanism of hormone action on the cell surface. Change in membrane permeability is a very important point. The receptor proteins of some hormones are large transmembrane intrinsic protein acting as ion channels for the facilitated diffusion of Na+, K+, Ca2+, these ions. On binding with specific hormones, these receptors proteins undergo conformational change so that membrane permeability for the ions is altered, resulting into important changes in the metabolism. The receptor proteins, there are some receptor proteins of some hormones which are large transmembrane intrinsic protein which are acting as the ion channels, ion gated channels for facilitated diffusions like Na plus, K plus, Ca2 plus. On binding with specific hormone, when these receptors or receptor proteins they bind with specific hormones, the receptor proteins undergo some three-dimensional conformational changes so that membrane permeability of ions is increased. Okay, it is altered, it is increased, resulting into the important changes in the metabolic pathway. For example, insulin promotes the entry of glucose from blood into the muscles and the cells by increasing the permeability of sarcolemma to glucose. The insulin promotes the entry of glucose for blood from blood into the muscles by increasing the permeability of sarcolemma, which is the membrane of the muscular fiber, okay, for glucose. The moment the permeability of glucose for sarcolemma is increased, then sarcolemma it can allow the glucose molecules to enter inside the muscular cells. Mechanism of action of hormone inside the cell, especially the steroidal hormones, they act within the cell, means intracellularly. They have intracellular or cytoplasmic receptors. Their small lipid-soluble molecules pass through the cell membrane and bind to the specific receptor molecules present in the cytoplasm. The receptor molecules carry them to the nucleus. Here, the receptor hormone complex binds to a specific receptor site on a chromosome and activates certain genes that were previously repressed or suppressed. Okay. The activated gene transcribe messenger RNA which directs the synthesis of enzyme protein molecule into the cytoplasm. The enzyme molecule promote the metabolic reactions in the cell. In the cell. Mechanism of hormone action inside the cell. You can see the hormone estrogen. These are the steroidal hormones. Thyroxine also work this way. These are the, for example, estrogen. They have uterine cell membrane. They come across the uterine cell membrane, they bind to the receptor present in the cytoplasm. Okay, the receptor hormone complex bind to the particular DNA site, which further initiates the formation of messenger RNA, which further form proteins, physiological responses, tissue growth, and differentiation. Okay, you can see the DNA proteins they bind to the receptor hormone complex, leading to the formation of pre-messenger RNA, which further form mRNA. mRNA it further helps in the synthesis of protein, okay, upon ribosomes and produce a response. Feedback control of hormone secretion. What is feedback control of hormone secretion? The secretion of hormones depending upon the age, daily routine, health of body, physiological conditions of body, etc. So secretion of hormones is dependent upon various factors like daily routine, health of body, okay, physiological conditions of body. Besides the above factors, hormone secretion is also dependent on its own amount circulating in the blood. Very important. That is very important. Besides other factors, hormone secretion is also dependent upon its own amount which is circulating inside the blood. Decrease and increase in the circulating amount of the hormone has a directly or inverse effect on the secretion of hormone. This is known as pull or push or feedback control mechanism of hormonal secretion. The optimum amount of hormone is very important in raising a particular effect, okay? Whether it is inhibitory or stimulatory. Feedback mechanism. Hypothalamus, okay, depending upon the low blood thyroxine levels, if the thyroxine levels in the blood are low, it will further activate hypothalamus. Hypothalamus, it further will release TRH, thyrotropic releasing hormone. Thyrotropic releasing hormone further activates the pituitary. Pituitary activation further increases high amount of thyroid stimulating hormone. This is positive feedback. If the blood thyroxine levels are already high, then it will further inhibit the hypothalamus or activate the hypothalamus to release inhibitory effects, or it will further inhibit the hypothalamus to produce less amount of thyrotropic releasing hormone, which further deactivates anterior pituitary or further signals anterior pituitary to produce less amount of thyroid stimulating hormone, which further deactivates the thyroid gland. Okay, so this is the negative feedback. Okay, negative feedback control is the direct feedback control. Thyroid stimulating hormone stimulates the thyroid gland to secrete thyroxine hormone. A high amount of thyroxine in the blood exerts an inhibitory effect on the pituitary to secrete less amount of TSH. This essentially results in a decrease in thyroxine levels. This is called direct feedback control. So negative feedback control is a type of direct feedback control. Now positive feedback control. Oxytocin released by posterior pituitary gland stimulate the contraction of the uterus during childbirth. As the contraction of uterus progresses, more and more amount of oxytocin is released. This is called positive feedback control. Oxytocin released by the posterior pituitary gland stimulate contraction of the uterus after childbirth. As the contraction of uterus progresses, more and more amount of oxytocin is released. This is called positive feedback. This is a general information about the endocrine glands, their origin. Pituitary is ectodermal in origin, pineal ectodermal, thymus ectodermal, thyroid thymus endodermal, thyroid endodermal, parathyroid also endodermal, adrenal cortex mesodermal, adrenal medulla ectodermal. Adrenal medulla is ectodermal and adrenal cortex is mesodermal. Okay. Testis mesodermal, ovary mesodermal, pancreas endodermal. So these are the different, origin or of these endocrine glands, especially the adrenal medulla, which is ectodermal in origin, and adrenal cortex, which is mesodermal in origin. Parathyroid, endodermal, thyroid, endodermal, thymus, endodermal, pineal and pituitary, ectodermal. Okay. Testis, ovary, mesodermal, pancreas, endodermal. Pituitary, anterior pituitary releases seven hormones. Hypothalamus releases two hormones, inhibitory and releasing hormones. Pineal gland, two hormones. Thymus, three. Thyroid, two hormones. Parathyroid, one. Islets of Langerhans, three. Adrenal cortex, 46. And medulla, two hormones. Testis, one hormone. Ovary, three hormones. Placenta, two hormones. Kidneys, two hormones. Stomach, one hormone. Duodenum, five hormones. And ileum is two hormones. Now comes the pituitary gland of hypophysis. Pituitary is known as hypophysis cerebri. Its name pituitary was given by Vesalius. Mueller's gland of Amphioxus and subneural gland of Hardmania is homologous to pituitary of vertebrates. As the vertebrates, they have pituitary gland. Amphioxus is having Mueller's gland, and Hardmania is having subneural gland. So Mueller's gland in Amphioxus and subneural gland in Hardmania is a classical example of homologous to pituitary of vertebrates. Weight of pituitary is 0.5 grams. Removal of pituitary is known as what? Hypophysectomy. Position and origin. Pituitary gland is the smallest, about 1 to 1.5 centimeter in diameter, endocrine gland of the body. It is pea-shaped, ovoid, reddish-brown gland situated at the base of the brain in a cavity, hypophyseal fossa of the sella turcica of sphenoid bone. Sphenoid bone is having a depression which is known as what? Sella turcica. This sella turcica is further having a depression which is known as what? Hypophyseal fossa. So pituitary gland is also known as hypophysis cerebri. Its name pituitary was given by Vesalius. Mueller's gland of Amphioxus and sub neural gland of Hardmania is homologous to pituitary of vertebrates. Weight of pituitary is 0.5 grams. Removal of pituitary is known as what? Hypophysectomy. Position and origin. Pituitary gland is the smallest, about 1 to 1.5 centimeter in diameter, endocrine gland of the body. It is pea-shaped, ovoid, reddish-brown gland situated at the base of the brain in a cavity, hypophyseal fossa of the sella turcica of sphenoid. It is connected by a short stalk called infundibulum to the ventral wall, that is hypothalamus of the diencephalon. So basically, pituitary is connected with the help of infundibulum to the ventral wall, that is hypothalamus of diencephalon. That is why it is called hypophysis cerebri. That is why pituitary is known as what? Hypophysis cerebri. Its weight is 0.5 to 1 grams. It controls most of the endocrine glands and it is called leader of the endocrine orchestra or master gland. Pituitary gland is closely related to hypothalamus, hence it is known as what? Hypothalamic hypophyseal gland. Pituitary is ectodermal in origin. These are the parts of pituitary gland: adenohypophysis and neurohypophysis. Okay, pituitary is ectodermal in origin. Adenohypophysis further having three parts: pars distalis, pars tuberculis, and pars intermedia. Okay, so adenohypophysis further having three main parts, major parts: pars distalis, pars tuberculis, and pars intermedia. That it all these parts make 75% of the anterior lobe of the pituitary gland. Okay, neurohypophysis contains parts nervosa and parts and infundibulum. Neurohypophysis or the posterior pituitary is mainly contained of parts nervosa and infundibulum. Posterior lobe 25%, anterior lobe 75%. So anterior lobe pars distalis, pars tuberculis, and pars intermedia. Okay, and posterior pituitary pars nervosa and infundibulum. Structure of the pituitary gland. Pituitary gland is comprised of two main lobes: adenohypophysis, anterior pituitary, and neurohypophysis, posterior pituitary. Adenohypophysis arises as the hypophyseal or Rathke's pouch from the dorsal wall of embryonic stomatium. During the embryological development, the stomatium, that is former mouth, it makes a deep elevation which is known as Rathke's pouch from the dorsal wall. This Rathke's pouch or hypophyseal, you know, we can say elevation is known as what? Is having this adenohypophysis. It is the anterior lobe of the pituitary. The neurohypophysis of pars nervosa or posterior lobe forms an outgrowth from the infundibulum, which makes the outgrowth from the infundibulum of the floor of the hypothalamus. So this is pars tuberculis, pars intermedia, pars distalis. This is the pars nervosa, posterior pituitary, infundibulum stem, and median eminence. Okay. So it is the anterior lobe of the pituitary. Neurohypophysis pars nervosa posterior lobe forms an outer growth from the infundibulum of the floor of the hypothalamus. Cells of pituitary. Chromatophore cells found in the adenohypophysis of pituitary. These are not stained by acid or basic dye. Pigment granules are absent. They are colorless, may change into chromophylls. So chromophore cells found in the adenohypophysis of the pituitary, they are not stained by either acid or base. Pigment granules are absent. They are colorless and may change into chromophylls. Chromophyle cells They are found in adenohypophyses of the pituitary. They are stained by acidic and basic dye. Pigment granules are filled in these cells. They are made of two types. Acidophils, it is known as cells synthesizing the and secreting the growth hormone and prolactin. The acidophils, it also known as the cells which synthesize and secrete growth hormones and prolactin. Basophils, these are known as what? Cyanophils, or cells synthesize and secrete TSH, thyroid stimulating hormone, and ACTH, adrenocorticotropic hormone, follicle stimulating hormone, luteinizing hormone, and melanocyte stimulating hormone. Okay, so basophils or cyanophils, they are the cells which synthesize and secrete TSH, ACTH, FSH, follicle stimulating hormone, luteinizing hormone, and melanocyte stimulating hormone. Okay, pituitary is the master gland of the body. It controls all the other hormonal secretions by feedback mechanism. Okay, now pituitary cells. These cells are found in neurohypophysis of the pituitary. They are supporting neuroglial cells and give support to herring bodies. So pituitary cells, these cells found in neurohypophysis of the pituitary, these secrete supporting neuroglial cells and give support to herring bodies. Herring bodies are dilated terminal portion of neurosecretory axon consisting of hypothalamo-hypophyseal tract system. These are hormone precursors for oxytocin and vasopressin. The chromatophore cells, chromatophilic cells, acidophils, basophils, pituocytes, and herring bodies. They are the six categories. Herring bodies are dilated terminal portion of neurosecretory axon consisting of hypothalamo hypophyseal tract. These are the hormone precursors for oxytocin and vasopressin. This is the blood supply, which includes hypothalamus hypophyseal stalk system. Okay, these are the neurosecretory cells of hypothalamus, herring bodies, pituocytes, neurohypophysis, axonal terminals. ठीक है. Blood supply to pituitary or hypophyseal portal system. I will be taking this part in the next lecture of the blood supply to pituitary or hypophyseal stalk system. Till then, bye bye. Take care. Stay safe. Stay healthy, everyone.